Okay, thank you, Viviana, for the kind introduction and for having me. Greetings, everyone from Argentina. We're having an unusually warm winter here. It's 32 degrees Celsius, something like 90 Fahrenheit. So greetings from the south. One of the nice consequences, one of the few nice consequences of the pandemic is that we can be closer and I can be showing you some of my recent work. So I'm going to be talking about a paper that is coming out on August 10th. So we're really excited to give you an advanced preview of some of the results you're going to see in the paper. And this is work that I did during my postdoc working with Mattia Lengyel and Guillaume Hennequin at CBL in Cambridge in collaboration with Lawrence Aitchison. So the motivation for this work is twofold, or the kind of questions that we're trying to answer are come from two different sides. And one is, how is the brain able to deal with perceptual uncertainty? We live in an uncertain world. I'm sure you're all aware of the fact that we often need to operate in conditions of low light, and when we see something and we care about a given object, we don't usually see this object floating in isolation in space. It's often occluded by other things, and stimuli and things we observe can be ambiguous. And we need to deal with this kind of uncertainty. We need to deal with a world where information is often noisy or incomplete. And the brain is actually pretty good at doing this. And I guess we became really aware of how good we are at these things when people started trying to train artificial systems to do the kind of things that we often do and we often give for granted. So there's more and more evidence that when faced with uncertainty, the brain is actually able to represent and operate with probability distributions to at least approximately, and in some contexts, perform probabilistic inference. So what do I mean by that? So let's say we have, we're dealing with uncertainty, so we're going to work within the realm of probabilities. And so if we have a probabilistic generative model of how sensory information is created, that tells you, well, if the things you care about, which are out there that I'm going to call my latent variables, have a such and such configuration, then what's the probability of observing a given stimulus? We define our model of the world by this probabilistic generative model. And then the thing we actually care about is the inverse of this relationship. That is, what is the probability of the things I care about, given that I have observed a given stimulus? And so in that case, Thomas Bayes comes to the rescue and tells us that if we want to compute this probability distribution, which we call a posterior, I'm going to be talking a lot about posteriors today. And if you remember anything from this slide, you should remember that this posterior means this probability distribution that of something I care about in the world, given the stimulus that I have observed. And we know how to compute it. And we also know that when we are faced with uncertainty in a probabilistic setting, this is actually the optimal thing to do. And so the brain can do that, apparently, but then what is the strategy employed by the brain to do that? And how is this implemented by neural circuits? How is this form of computation carried out? How are these probabilities represented in the brain? So this is going to be our first question. But then, on the other hand, from the perspective of neural circuits and cortical dynamics, what we want to find is a common framework for a variety of features that tend to appear again and again across different experimental conditions and across different species, which is the fact that when you present the same stimulus, even the same constant stimulus, neural responses tend to vary, both over time and across trials. So we have some raster plots of spike trains here, and we see that neurons tend to fire a little bit differently every time. So as you know, historically, or in the beginning in neuroscience, people saw this as a signature of, well, noisy, imperfect computations in the brain, and one way to get rid of this noise is to take averages, right? So you could take population averages, or you could take average over time to get rid of this kind of variability. But it turns out that there is valuable information encoded in this variability about the stimulus. So these might not be all garbage to throw out, right? So some of these systematic variations and which contain information about the stimulus are transients. So we see that neurons tend to fire higher at the onset of the stimulus and then decay to a resting response value. The size of these overshoots encodes information about the stimulus. So here we see that if you present the same image at different contrast levels, then the size of the overshoot will tell you something about the contrast of that image. If we look at the frequency domain and you look at different frequencies, you see that there tends to be a strong peak in the gamma band. So we have gamma oscillations. And the exact frequency of that peak is also not constant, but it is shifted with the contrast of the stimulus. So again, these gamma oscillations are stimulus dependent. And finally, if you look at variability, we know that variability tends to be quenched at stimulus onset. So the stimulus reduces the amount of variability in the system. And moreover, the way that variability changes is also tuned to the stimulus. So all of these things are telling us that there is more information being encoded in the variability of neural responses. And of course, there have been a number of theories to try to explain these elements individually. But the question we're asking here is there a unifying framework? Is there a way to bring all these things together under one theoretical umbrella? So this is going to be our second motivation for this work. So the proposal is as follows. What we're going to do is we're going to optimize a recurrent neural network that respects a series of biological constraints, because we're going to try to capture something about dynamics in the brain. And we're going to train it for a computational objective, that is sampling-based inference. We're going to train it to inference to compute these posterior distributions I talked to you about in the beginning. And wait for me just a little bit. I'm going to talk about why sampling-based and what I mean by that. But for now, what I'm going to tell you is that we're going to use machine learning tools to train this artificial neural network that shares some properties we think are important from the brain for a computational goal. And then once we do that, we're going to open this black box and look at what happens with neural activities in the network while it is solving the task. And we're going to see if just by training a network with these characteristics to achieve a certain computational goal, these kind of properties we see in the brain are somehow recovered. So let's get started. As I said before, the first thing we need if we're going to talk about inference is a generative model. That is a probabilistic model of the world as far as these, as the task that we care about is concerned. So in this case, we're going to take a very simple visual task where our latent variables are going to be orientations. We want to infer the content in terms of orientations in a small patch of an image. So each of our latent variables is going to represent an orientation. So we are going to find the probability distribution to observe a given patch by taking these latent variables and projecting them via some Gabor filter projective fields. So this is our generative model of the world as far as the task is concerned. And then, as I said before, the optimal thing to do if one receives a stimulus like this, an image patch, is to invert this probability relationship via Bayes' rule to obtain the probability of latent variables or orientations taking certain configurations given the stimulus we have observed. We're going to call this the ideal observer because this is as best as you can do. So what are we going to ask the network to do? So we're going to ask the network to mimic what an ideal observer would do in the same scenario. So we take a ring of neurons, and you notice the similarity between the structures here and here, because we're going to map each excitatory cell to one of the latent variables in the model, and we're going to have additionally inhibitory neurons that are going to be auxiliary units in the system. We're not going to specifically constrain the activity of inhibitory neurons, so that when the network receives the same image patch as the ideal observer, it reproduces the same posterior probability distribution predicted by the ideal observer. And in just a second, I'm going to tell you what do I mean by reproduce that distribution or encode that distribution within its activity. So I mentioned the word sampling-based inference, and what do we mean by that? So let's say we have a stimulus and a small image patch. This is all the cartoon. We are actually thinking of much smaller image patches, but just to illustrate. And so the ideal observer takes that stimulus and says, okay, I'm going to use Bayes' rule. And with that, I give you a posterior probability distribution, and it looks like this. This is my posterior. That is the probability that given certain orientations take particular configurations given the stimulus I have observed. And here I'm just showing a 2D projection of this distribution. We have more directions in space, actually. At the same time, I'm saying, okay, I'm going to feed the same patch to the network. And this is going to be a stochastic recurrent neural network whose activity is going to evolve over time. So if I plot the neural activity of two cells here in space, what I want to do is I want to match the distribution of neural activities here to the distribution given by the posterior. That is, as the activity evolves over time, if I collect enough samples, I'm going to reproduce or I'm going to encode this probability distribution in the activity of these neurons. And this should happen not just for one particular stimulus, but for any possible stimulus that I present. And you notice right away that when you change the stimulus, this probability distribution is shifted, but also its width changes and its overall shape is changing, which means that neurons in our network are going to have to not only modulate its mean responses, but also its structure of covariability, right? And so this is making an important assumption, because what we're saying here is that the mean of the activity is going to be, well, if it's a Gaussian, it's going to be related to the position of the peak of this distribution. And that is, the mean is going to tell us something about what's the most probable configuration of variables. But variability in the network is going to be directly encoding uncertainty in the probability distribution, because the width of this distribution is going to be related with to how uncertain are we about what we're observing, right? So in this case, uncertainty is going to be encoded directly by variability in neural responses. So we want to equate these things for every possible stimulus, and I'm going to be a bit more concrete now about the model, both the generative model and the network. So this generative model that we're going to use is called the Gaussian scale mixture model. And we're using this model because it captures the statistics of natural images quite well. And basically, an image patch here is produced by a certain combination of orientations. And then the whole patch is globally scaled by a contrast variable. So we can modulate the contrast of the patch. And so the nice thing about these models is that posterior probability distributions are Gaussian, and this is going to simplify our analysis and allow us to optimize these networks much better. And from the side of the network, the model we're going to use is the stabilized supralinear network. We have the equation here, so it's a very simple rate-based model where we have a leak term, we have our recurrent connections coming here, our input, and some noise that is going to be stimulus independent. And you notice here that if we call U our membrane potentials of these cells, that these are not connected directly via membrane potentials, but we have a nonlinearity here, a rate function. And for this nonlinear transfer function between membrane potentials and rates, we're going to use a threshold power, which is the kind of thing that has been observed in cortex. We're not going to use a ReLU like people usually do in machine learning. And we're also not going to use a sigmoid. While we think neurons, of course, have a maximal firing rate, it turns out that they usually don't operate in that regime. So we're going to use more realistic transfer function. And you can imagine that if you have a recurrent network with a nonlinear explosive excitability like this, then this can become quite easily unstable. So inhibitory cells in the network are going to have to get tuned in a certain way to be able to keep the whole thing stable at all times. We're going, as I said, the posterior distributions are going to be Gaussian from the model, and we are going to approximate neural activity as being Gaussian as well. And if that's the case, in order to match the two distributions, all we need to do is match the first and second order moments. So the mean and the covariance are going to have to match. So our goal is going to be to learn the recurrent weights, the structure of noise covariance, which is going to be constant, it's not going to be stimulus dependent, and some other parameters of with which we transform the input before it enters the network. And the goal is going to match the first and second order moments for every possible image. And so compare here to what is standard when people are trying to train neural networks, which is usually to try to just match mean responses to some target values. So here we're going to have to match not just the mean, but also the second order moments as well. I'm not going to go too much into detail of what we do from the machine learning side, but just for you to know what's a bit under the hood. We are able to evaluate how neural activity evolves over time in the network. Whenever you can simulate an experiment in which an animal suddenly sees a stimulus as they would, and then you can simulate neural activity in the network, compute these moments, and then what you try to do is get those moments to be as close as possible to your target moments given by the posterior distribution, and then you take another image and you repeat. And additionally, we're going to penalize temporal correlations in the network, since if you want to represent a distribution using samples, then you better do it fast, because otherwise time is a problem. If you need to take a very long time to be able to represent anything, then this is not going to be useful in any real life setting. So this is our cost function, and then we use backpropagation through time to optimize the network for these computational calls. So how do we do? So in order to be able to tackle this problem successfully, we've had to assume a certain symmetry in the problem. Given that orientations are on a ring, we assume circular symmetry, and that allowed us to train actually a very small number of parameters. And also it allowed us to use a very small training set in order to train the network. So we actually used only 5 images, which is like, it's actually one image at 5 different contrast levels. So for each of these images, the ideal observer tells us, this is your posterior distribution, this is your posterior, this is your posterior. And so we have our inputs and we have our targets, and we train the network for that. These are the connectivity patterns in the network. We have these kind of smooth patterns that decay as so if 2 cells have different, the more different the orientation preference, the lower the connectivity between cells, and we learn the width and the height of these profiles, and something very similar for the noise covariability patterns. And so we compare now the mean responses, these are population averages, and the standard deviation, that is the square root of the variance in the network, for the posterior distributions in green, and the network results in red, and we see we get a pretty good match for these 5 different contrast levels. If we look at actually the mean and the standard deviation of each cell, which corresponds to each of the possible orientations in the posterior, we see that we have actually a very good match between the distribution of network responses and the posterior distributions prescribed by the ideal observer. And this is all fine and good, but these are only 5 images. So what happens if we now take arbitrary image patches? Are we still doing a good job? So the first thing we tested was, the easiest form of generalization here is to evaluate what happens with intermediate contrast levels. And we see that the network smoothly interpolates between the values that we have shown, so there is no crazy overfitting going on. And then we just generated a lot of random image patches, some of which contain more than, so if we look at the image here, we see that there is a single dominant orientation. But when we generate random patches, you obtain patches that have more than one dominant orientation, as we do here, and the network is able to perform really well even in these settings. So there is some degradation of the performance, but overall, we show that the network is able to actually perform inference for a wide variety of image patches. In the beginning of the talk, I told you that, okay, we're going to use a machine learning method to train this network, but at the end of the day, what we care about is what happens in the brain. We care about neuroscience, and so the big step was to now, once you have a system trained, to open up this black box and to look at what is going on with neurons while they solve the task. So that is what I'm going to show you next. We were really excited and surprised to see these results. So here on the left, I'm showing you experimental results. And on the right, the results from our network. Most of the experimental results come from awake macaque V1, except for these ones at the bottom, which are coming from mice. So the first thing we checked was that the tuning curves in the network, in terms of firing rates, I didn't really say this before, but remember we have membrane potentials and we have firing rates in the network, and we did moment matching for membrane potentials. But also when you look at firing rates in the model, it turns out that the tuning curves that we observe for firing rates are quite reasonable, and they are also within the same range of activities as you see experimentally. We also observe variability quenching. So here in red, we have spontaneous activity, and in orange, it's evoked by the stimulus. We see that variability is reduced as the stimulus is presented, as it happens experimentally, and that the shape of this variability quenching is also quite similar. We see that cells, if you present a stimulus, then cells whose preferred orientation is aligned to that stimulus will reduce their variability more, as it happens experimentally. But all this is still something that has to do with the overall stationary distribution of responses. There's not really, the issue of time and dynamics hasn't really entered here yet. What we were surprised to see, because the training of the network only involved the stationary distribution, and regarding time, all we said was do it fast, but that's it. What we were excited to see was the fact that we observed gamma oscillations in the network. And not only that, but if you notice in this plot here, we see that, as I mentioned before, the peak of that gamma frequency, it shifts when you change the contrast of the stimulus. We knew that, and so we see here experimentally this effect again. So the gamma peak shifts to higher frequencies as the contrast increases, and exactly the same happens in our network. We are able to measure the peaks of these within the gamma band, and we see exactly the same effect. Not only that, but the network displays these kind of realistic transients with overshoots, which also scale with contrast in a very similar way as it happens experimentally. And these transients are inhibition-dominated. That is, there is a transient imbalance between excitatory and inhibitory conductances, and that is dominated by inhibition, as people see experimentally. None of these things were directly encouraged by the training objective. They arose indirectly. So one of the first questions that we had was, well, is this inevitable? Is this just an inevitable consequence of the way we have proposed the way we have written the model, the choice of, I don't know, membrane time constants, the choice of the structure, and so on. So we had to rule out that possibility. So the first thing we tested was to do the exact same type of training, but for a different objective. So we proposed these control experiments in which you train the exact same model, and you take the same targets, but you ignore the second order moments of the distribution, and you only train for mean responses, as people have classically done many times with other networks, right? So this would be like the classical thing to do. And when you do that, we observe that I'm not showing gamma peaks here because there are actually there's no gamma peak in the spectrum, so we cannot find the maximum. So gamma oscillations are abolished. We find only very weak transients, and it's hard to talk about inhibition-dominated in this case. So it seems that all these dynamical features that we see, that we observe in the model, are quite specific of the particular computational goal we have trained the network for. And I'm not showing it here, but we have also trained the network to match mean and variances, but not covariances, and these things don't appear either. And we also have trained the network for the same task, but trying to enforce final factors to stay constant, as you need with other probabilistic frameworks. And then these things are abolished as well. So it really seems to be quite specific of the goal we have proposed. If we look at mean responses in the trained network, so let's say you came across this optimized network and you don't know what it does, and you only look at mean rate responses, then you find out that everything the network does is just divisive normalization, which is a canonical model for computation in the cortex. So the model does recover that, but it was not really trained to do that. Okay, so far, all of these results are, all of these experimental features were previously known, right? And what we show is that this model is able to capture all of these things. What I want to show you now is some of the new things we have learned, some of the new predictions of the model that we have been able to go out and test. So we thought long and hard about why transients and oscillations appear in this network. So it is clear that they're useful because, as it happens with optimization, if something is there, it's because it's useful. Otherwise, it wouldn't emerge. But then why? I'm not going to be able to talk about oscillations here today, but you can read that in the paper. So I'm going to focus on transients here. And so what we saw was that these transient overshoots speed up sampling on the fly. That is, if you have to do something very, very quickly and immediately after the stimulus comes on, then you need these kind of overshoots to be able to do it well and fast. So the idea is as follows. I'm just going to briefly sketch the idea behind this. And the idea is, let's say you have to follow a signal that changes over time. In this case, the mean from the posterior distribution. So at some point in time, the stimulus comes on. So you have to stop tracking your spontaneous distribution, and you have to move to the distribution described by the posterior corresponding to the stimulus. And if you're trying to represent this mean using a finite number of samples, so you have a trailing window to compute an average, then this average can be thought of as a convolution between your neural responses and an averaging kernel. And if what you want to do is match a target that changes over time, and we ask the question, so what is the smooth neural signal that minimizes the distance to that target. And we're saying smooth because we think neural responses kind of instantaneously switch over time. And it turns out that you can do the math analytically for this simple problem. So the solution to this problem is the deconvolution of the signal subject to some smoothness penalty. And you can compute the actual result for that. And I don't need to pay attention to the exact shape of this formula, but what we see is that neural responses will have to be proportional in size to the mean that you're trying to match. But when you actually plot this solution, you see that the optimal solution is to have transient overshoots with ringing, as you see in the network. So what this analysis is telling us is that, okay, it makes sense to have overshoots because that is the optimal thing to do when you want to follow this signal. But it goes beyond that, because it is telling us that since overshoots have to be proportional to the mean you're trying to achieve, then if we look at the vector of neural responses in neural response space, then these overshoots have to be aligned with the change in mean responses. And this means that overshoots will have to be tuned to stimuli in the same way or in a similar way as neurons' mean responses. So this was a specific prediction from the model. Transient overshoots should also show stimulus tuning that is very similar to typical tuning of mean responses. So we looked at that. We also analyzed these numerically in the network. I'm not going to go into this part here, but indeed what we saw was that both so we saw that in our model, overshoots were indeed tuned to the stimulus in a way predicted by this analysis of the deconvolution. So we looked at the Ecker dataset that they were so kind to share with us, the same dataset that we used to analyze average firing rates and final factors when I did the comparison with experimental results. And we did a novel analysis of this dataset. To our knowledge, people had not looked into this before. And we indeed found that overshoots themselves were tuned to the stimulus as it happens in our network and as predicted by this optimality principle that I just mentioned. So even if we plot overshoots versus the steady state difference, there is the change in mean responses for individual cells and individual experiments, we see that they are quite well correlated. So this was an entirely novel prediction of the model that we were able to verify experimentally afterwards. So in summary, we were able to train a stochastic, fully recurrent neural network respecting Dale's principle and employing a realistic, nonlinear input-output function to perform fast sampling-based probabilistic inference. We found that the network correctly generalizes to novel stimuli and that their static responses show divisive normalization. And we show that this network that was trained purely for a computational function that involved steady state responses actually featured a number of dynamical features that are typical of cortical circuits, namely stimulus-dependent gamma oscillations, strong inhibition-dominated transients at stimulus onset, as well as variability quenching. We went beyond that, and we were able to show that the model produced new predictions regarding the tuning of transients, and we were able to confirm these in a novel analysis of experimental data. So we think what's nice of this is that it brings a wide range of experimentally observed phenomena under a single theoretical framework, that is that of a sampling-based inference. I have a bit of time, I think, so I was not planning to show you the results regarding oscillations, but since I have time, I'd like to show a bit of that. So before I show you this, so similarly to what happens with transients, one can study why oscillations emerge in the network. What function do they play in sampling. And so what we did first was to study a very simple system, a 1D system, where the goal is, again, taking your traces of neural activity to match a given distribution using a finite number of samples. But in this case, instead of looking at what happens at stimulus onset, we think that we are already in the stationary regime where neural activity is just fluctuating and the stimulus has been on for long enough. And so we simulate different traces of neural activity where we can switch on and off these oscillations or make them stronger. And then we can compute the difference between the implied distribution by these samples and the target distribution measured by something called the KL divergence. And we see that indeed having oscillations allows you to converge faster to the target distribution than if you didn't. So if you're collecting samples over time, you're going to better represent your distribution if you're using oscillations that if you're not. And a way to think about that is that while the stationary distribution is always going to be the same, in the absence of oscillations, you're just going to be usually sampling your distribution more locally. And these oscillations allow you to traverse longer directions in state space faster so that you get a better estimate of the distribution. So once again, if this is the reason why you observe oscillations, then as it happened with transients, you expect to find these oscillations where they matter the most. So if we take each posterior distribution, now in the full network and the posterior from the generative model, and we project neural activity into the different principal component directions, that is, the directions that capture most of the variance, we see that indeed the network is, and we plot here the power spectra for projecting neural activity along these different directions. We see that gamma oscillations are indeed stronger along the higher principal components of the distribution. That is, this network is using these resources, which are oscillations in the directions that matter the most, which are the ones that explain most of the variance, and less so in the directions that don't explain so much of the variance. And this is another prediction of the model. This one we haven't tested yet, but as you see, this model allows you to now formulate a bunch of new predictions and study a bunch of new things, which we think is really nice. So now, with that, I would like to give a special thanks to Alexander Ecker, Philip Behrens, Matthias Bethje, and Andreas Tolias for kindly sharing their data with us, to our founding sources. And I wanted to briefly show the institute where I'm currently based at, which is the Research Institute for Signal Systems and Computational Intelligence, located in the city of Santa Fe. We're here on the top floor here of this building. This is the city of Santa Fe. People in the institute, I was actually not there yet when they took the picture, so you're not going to find me there. And with that, I'd like to thank you for your attention, and I'll be happy to take questions now.